And this observation was then basically used to develop a new strategy to eventually also generate full-length dystrophin, or at least or the truncated version of dystrophin, I should say. Okay. The idea behind this is that we have, as I said, 79 exons that are encoding dystrophin. And what is symbolized here is how these different exons have different reading frames. I'm sure you know that there are different reading frames. You have these triplets, and depending on when you have, for example, a point mutation and you fall out of this reading frame, then you will change the reading of the mRNA, and eventually you're gonna run into a stop codon because you're out of reading frame, and then the ribosomes falls off the message. And this is basically depicted here, the different reading frames. You could imagine this is one reading frame, there's another reading frame, and for example here, this is the third reading frame that is possible. And you see, depending on the splice junctions between the exons, exons can have different reading frames. And it's important that the reading frame is, of course, conserved so that you can then end up with a full-length protein. Now, in Duchenne patients, it may happen that you have, for example, and this is generated by splicing, as you all know. This is just not really that important, that you have to get rid of the intronic sequences here, and then you splice the appropriate exons to each other, so that you then get this open reading frame and you get the dystrophin protein. Now what can happen is that you have, for example, a deletion of some of the regions that encode certain exons. In this particular case, there is a deletion that gets rid of exons 48 to 50. And then if you lack this and splicing is ongoing, then exon 47 would splice into exon 51. And if this happens, you change the reading frame. What then happens is that you generate an mRNA, a mature mRNA, where now exon 47 is next to exon 51. Because the reading frame does not fit here, what then happens is that protein translation will stop, because you will be out of frame, and eventually, by chance, you're going to hit a non-coding a stop codon, and then as I said, the ribosomes will fall off and the translation will stop, and you end up in a situation like in a Duchenne patient, that you basically can't anchor the extracellular matrix, which is here symbolized with this ship here, to the F-actin cytoskeleton. And then you end up having a truncated version of dystrophin that is non-functional. Now, when you identify these mutations, you can, for example, eventually generate a method, and that's the aim of this exon skipping, that you get rid of, or that you prevent splicing in of exon 51. If you were to do this and you get rid of exon 51, then you generate a full-length or a partial transcript of mRNA that now, where exon 46 and 47 then is spliced into exon 52 and and then followed by exon 53, and as you can see here, the reading frame would be conserved so that the protein can be translated, the ribosomes don't fall off, although the protein is a bit smaller, but then dystrophin is still anchoring here, this ship here, to the anchor. I mean, in symbolized, the extracellular matrix is still connected to the F-actin cytoskeleton, although the the protein is a bit shortened, but this does not really matter for the function. And this is exactly the strategy people have developed, and they have basically developed so-called antisense oligonucleotides that are just small oligos that then sit at the splice junction and eventually try to prevent the splicing of the splicing in of exon 51. If this antisense oligonucleotide prevents this, then you will end up, as I said here, with this message that where the open reading frame remains. Of course, this method, this antisense oligonucleotide you design, is very specific for a particular mutation. For each patient it would be different, and here this is what you see here is a calculation of how many patients of the entire Duchenne population would benefit from a skipping of exon 51. As around exon 51 there seems to be some hotspot of mutations. The largest percentage of the patients that would benefit from skipping exon 51 is 13% of all the Duchenne patients. That's why companies have at the beginning developed antisense oligonucleotides that target here exon 51. And there are basically now several chemistries available to do this, because of course you can imagine that the challenge of this is that these antisense oligonucleotides, because they have to act on pre-mRNA, they need to enter every nucleus or most of the nuclei in a fiber, and they have to then sit on this pre-mRNA, which is of course quite a challenging aspect of how you try to develop these antisense oligonucleotides. Okay, there are basically two chemistries of this. One that was originally developed by a company called Prosensa, another which is similar to these morpholinos one uses to interfere with zebrafish, in zebrafish with gene function like an antisense oligonucleotide. These are modified morpholino-like nucleotides or short oligonucleotides that seem to be more stable than this regular here 3'O methyl phosphocyanate modified oligonucleotide. It turns out both of these companies then targeted this exon 51. As I said, this is basically the mechanism. If you have an oligo that sits on a splice junction, you can prevent the inclusion here of this particular exon B, and which would be in this case exon 51. And this then prevents the inclusion of this, and therefore you have then a splice version which is a bit smaller, but as I said, keeps the reading frame open. Both of these companies have run clinical trials. This Prosensa trial here failed. The problem with this was that the oligo was not really very stable, and also the patients, they got some issues at the injection site. They get a very strong reaction in the skin, and they also got some problem with the kidney. This has then been stopped and didn't really work. In contrast, this oligonucleotide, this morpholino type, they had at least some partial benefit, and I will show you this briefly, what the data are that have then led to the conditional approval of this oligonucleotide, at least in the US. It's not approved here in Europe. Basically what they did, and you see here, that's one of the issues of course with these rare diseases, that you don't have that many patients that can enter a clinical trial. And in this particular case here, they run a so-called phase 2 trial, which is after the safety, the trial that in a set of patients tries to establish efficacy. They had 12 boys that were treated, 4 each with a lower and a higher dose of this oligonucleotide, and 1/3 of the boys was treated with placebo. That's another challenge in this field. What would be the primary readout, what you're going to look at? And they decided to look at, on the one hand, to look at expression of dystrophin. Of course, this is really the target of these antisense oligonucleotides, but they also tried to look at how far these Duchenne patients can walk in 6 minutes. This has become, although it's now also debated, but it has become at this time the gold standard for these neuromuscular diseases. I mean, you just measure how far the boys can walk in 6 minutes. Of course, if you want to measure this, you need boys that can still walk. In other words, your cohort is selected to boys that are pretty young. You cannot include all of the boys that already have lost ambulation, and the sensitivity, you can also imagine, sensitivity of this 6-minute walk is rather difficult, because see how far you can walk in 6 minutes is also very much depending on your motivation, how far you want to walk in these 6 minutes. One day you might be in a bad mood and you don't like to walk, or something hurts, you have a headache, and this all affects, of course, the 6-minute walk test. And this has really turned out to be quite a difficult test to also find consistency, because it's quite variable. And if you have only 4 boys, you can imagine it's really going to be difficult to reach this. But anyway, what they did, they had this placebo control, and then at a certain time point, after 24 weeks, they started to switch those boys also to the treatment control, to high and low dose of control, and at the end all of these boys were then treated with this drug. What you can see here, that after treatment, these boys start to really make this skipped mRNA, but this is RT-PCR. You can imagine RT-PCR is rather sensitive. You can detect even a low percentage of skipping you can detect with RT-PCR. And this here is now a Western blot, and you can imagine actually there is not really that much you can find in terms of dystrophin expression, because here this would be a healthy control, and this would be basically the loading. I mean, if the healthy control, the loading control is so little, is only shortly exposed, you get this huge amount of dystrophin, while you need to expose this loading control here so long that you start to see something in the boys, which means I think the efficacy is really a problem with this antisense oligonucleotide. But nevertheless, they then continued to treat those boys, and then they found, when they compared these now 12 treated boys with historical controls, this is basically the progression line over 36 months. This dotted line, is the line what boys lose in ambulation over 36 months. And you can see here, when they start at the 6-minute walk test of 350 meters, after 36 months, 3 years, they can walk only 100 meters after these 3 years. And in contrast, these 12 treated boys here, they kept the distance, was still over 250 meters, and this delta of 151 meters here is really significant, highly significant, as you can compare here with the historical control. The trouble with this is that this historical control is, of course, one does not know how well this is and how reproducible this is, and the numbers are still pretty low. The other trouble is that this historical control here is really only boys that are amenable to exon 51 skipping, because when you compare now historical controls of all of these Duchenne patients that were in this history trial. You see here that they have also other mutations, that they lose less than basically these exon skipping 51 amenable boys here, which is already a delta of 94 meters. In other words, if you were to take now all these historical controls here and you compare them to these 12 treated boys, you're not going to end up being a significant, not being significant here, that the treatment would result in a significant difference or improvement of the walking distance if you were to take these 36 patients here. Nevertheless, the company succeeded to persuade the Federal Drug Agency in the US to get approval. In other words, now these boys that are amenable to exon skipping 51, they can be treated with this drug. And they were conditionally approved, which means that they need to run clinical trials and also to see that in then the usage, how the walking distance develops in those boys. You should also, you can imagine that these are so rare diseases, and you have a lot of parents and patient organizations that really create a lot of pressure on these federal agencies to approve a drug, even if the efficacy you observe is still minimal. But nevertheless, I mean, these are so few patients that this is not a real big burden also for the health insurance, and that's why this has been approved. It has not been approved in Europe, as I said. The reason for this is that in Europe they have approved yet another drug, which is a drug which suppresses nonsense mutations, which in my view is not at all functional. But then once you have done this as an agency, and you realize that maybe this decision was rather thanks to the pressure of patient groups rather than because of really scientific strong data, then you start to be very cautious with the second case you have, because you already made such a mistake quote unquote, and then you get really more cautious. That's why I think you wait then until this other branch, let's say the US here, now finds out that whether this treatment really works. But I think it's nevertheless, I think in terms of the theory and the applicability, this, in my view, is really also going down to the basic mechanism of the disease, and I'm pretty convinced that it would work. The problem is just that these oligos really need to go into the nucleus, and they really have to sit at this splice junction. And they really have to prevent this splicing from happening. And this efficacy, of course, if you give it systemically, you can imagine it's not that trivial to have the drug reaching all these nuclei and then suppressing this. I mean, it's really an issue of how to deliver these oligonucleotides to the patients. But I think, as I said here, this is really the most promising way to do it. I think gene therapy, as well as these skipping methods, they are really have some promise. I should also mention that also in the spinal muscular atrophy, where this first gene therapy was approved, Zolgensma, that is now really also can be used for patients also in Europe. That is this expensive. Also in this spinal muscular atrophy, there is actually another company called Biogen that sells a similar idea with an antisense or with an oligonucleotide that has to be given intrathecally into the spinal fluid, that allows to express another gene that helps to compensate for the spinal muscular atrophy. Also, this treatment is quite successful. It's very similar to this antisense oligonucleotide idea. It's very successful. Also costs a fortune, but also has been approved both in the US as well as in Europe. It's the company that sells it is called Biogen. The drug itself is called nusinersen. You can maybe look this up. It's quite interesting also to read about this. Okay, just for this in general, and what I would like to take as a take home for you, that now we see that gene therapy with this microdystrophin has now become really feasible. I should also mention, by the way, Roche has bought the license for, in case this gene therapy with microdystrophin is approved from the company that does this is also called, is so-called Sarepta. They have paid 2 milliarden Swiss francs to have the license to market this treatment then within Europe, which is also quite a lot of money. I should also maybe say Novartis has bought the original company that generated this spinal muscular atrophy drug that is now sold for 2 million. They bought them about 3 years ago or 4 years ago. The company is called Avexis for $8.5 billion, which is 8 milliarden Swiss francs, which is really a lot of money for such a rare disease to be treated. But Novartis thinks that gene therapy is really the emerging way to treat many diseases. Okay, exon skipping and gene therapy, they really target the primary cause of the disease, which I think is really key in all of this, because you really have a higher efficacy. And of course, if you really are successful in increasing dystrophin, this should lead to a profound improvement of the disease, because this is also the cause of the disease. The advantage of gene therapy is that it can be applied to all of the Duchenne patients, while exon skipping can be applied only to particular diseases. It's really a very good example of so-called personalized medicine, because it's really depending on your mutation which type of antisense oligonucleotide you can use, or whether you can indeed actually use antisense oligonucleotide. The challenges that remain is that you really need to reach all of the muscles, that microdystrophin needs to be expressed also long term. If you think that when you inject AAVs into a patient, the patient will eventually develop antibodies against this virus, and then you cannot apply the same virus again in 5 years or so, because this virus will instantaneously be cleared by the antibodies that were generated by the patient. The trouble with the oligos is, of course, that you need really high efficacy, and you may also generate some side effects which are probably even worse than with AAV, because of the chemistry. And as I told you, there were some kidney issues with one of the antisense oligonucleotides. And I also mentioned penetration of the tissue is really an issue here, and that the other challenge is really that each oligo may eventually have, if it targets other exons, is a new drug, because it may also have other side effects, and this of course makes then development very slow and expensive, because you need to do all the toxicology before you enter clinical trials, and all this makes it really expensive and also quite slow. And the more oligos you generate, the less patients you can eventually treat. I told you exon 51 is the most frequent one. But if you were then to go down to where you have only 5% of the patients that can be treated, then it's actually the companies will calculate it's maybe not worth the effort to treat only such a few patients. The other issue, of course, is if you have a lack of dystrophin in the patients, if you then all of a sudden generate dystrophin, you may generate immune responses within the patient, because this patient has never really observed dystrophin. One argument against this is the presence of these revertant fibers. That's why one can argue, you still have these revertant fibers, and therefore the patient may have some tolerance to the dystrophin. And as I said, exon skipping, of course, does not allow to treat all of the patients. One other thing I would like to mention, that now, at least preclinically, there have now also been some attempts to use CRISPR-Cas9 editing for Duchenne muscular dystrophy in the muscle. I mean, there you bring, similar to the paper you read, you bring in AAV that expresses Cas9, and in with a second AAV you bring in, for example, a guide RNA that then makes small deletions and, for example, gets rid of exon 51, so that then genetically on the DNA level it would be removed, so that you would, for example, come again into the frame. You can imagine also here, of course, there are lots of challenges that this eventually works in patients. Now, this is basically it what I want to say to muscular dystrophies and therapies. Are there questions? Okay. Seems not to be the case. Then I start this other lecture on the neuromuscular junction, because it's the other important thing. You look also in your mouse groups, and I will also talk then at the end a bit on the maintenance of this structure, as well as how this structure changes and the mechanisms that might be involved, also what you study in one of your mouse groups on aging. What I would like to do is briefly explain you the development of the neuromuscular junction, and then I will try to explain some of the mechanisms that are involved during the development, and these mechanisms are also important for the maintenance of the nerve muscle synapse. I have shown you this, you are probably now aware of this, that the neuromuscular junction, and you have also seen it in the whole mounts, is really the contact point between the motor neuron and the muscle. And as I explained to you, and probably also have heard now several times, this is really an important structure, because it's destined to transmit the action potential from the presynaptic side to the postsynaptic muscle fiber, and for that it's highly specialized. It has a high concentration of acetylcholine receptors here at the clefts, and high concentrations of voltage-gated sodium channels at the troughs of the folds here. You also see that they have these myonuclei that are specialized, that underlie the synapse, and today you will see on this RNA fish experiments, you will actually see some of these genes, how they change their expression also during aging. The development of the nerve muscle synapse, that's the time frame in mice, is of course occurring in utero, and it starts already at embryonic day 11 to 13, when the nerve starts to approach the developing muscle fibers. I mean, at this particular time point, muscles already start to form so-called primary myotubes. They start to have these multinucleated fibers, and they also start to express acetylcholine receptors. And these acetylcholine receptors at the beginning, as you can see here, they also already end up in in primitive clusters when you look at this. And the nerve then starts to approach those developing myotubes and starts to generate also these neuromuscular junctions. This observation that these acetylcholine receptors are preformed has created quite some, how how should I call it, maybe discussion on the mechanisms that are involved in the setting up of these structures, because some people in vitro have shown that the nerve is really 100% required to set up these structures, while if some in vivo experiments at these early stages indicate that these clusters can already form in the absence of the nerve. I think one of the explanations for this is that these acetylcholine receptor clusters, they tend to cluster because if they're highly synthesized, they start to assemble postsynaptic like structures, and then it's the nerve that is required to stabilize them. And I will explain to you what is the stabilizing activity in the muscle. But anyway, I mean, after this initial touch here, these clusters become more and more associated to the presynaptic nerve terminal. At this stage here, at embryonic day 14 to 19, and the mouse is born around embryonic day 20-21, which is the end of the gestation in mice, you start to see here, and this is also the time point where you have the formation of so-called secondary myotubes, which are formed along the primary myotubes here, and which is important to create the increase in the size of the muscle. You see that the nerve is already present, and the acetylcholine receptor clusters are now quite closely associated to the presynaptic nerve ending. And then with time, you also, when the muscle starts to twitch, then you also start to see that these myonuclei, these fundamental myonuclei, become aggregated underneath the nerve terminal. And then you also start to lose any of these acetylcholine receptor clusters that are outside of this innervation zone here. Also, you start to see the development of a basal lamina or a basement membrane between the nerve and the muscle, and this will become then important in the later then in developing or identifying factors that are important for this formation. After the mouse is born, then Schwann cells, which are not the Schwann cells that isolate, that form myelin, but these are so-called capping Schwann cells, a specialized Schwann cell that sits on the presynaptic nerve terminal and sort of insulates it, and also participates with calcium signals to the action potential generated by the nerve itself. They are also coming and cap the nerve terminals. It also turns out that at the beginning, before the muscle, at birth, muscles are multiply innervated. In other words, there is more than one motor neuron sitting on the same site of innervation. And then with time, in the mouse, it's about 2 weeks after birth, all but one of these synapses or of these motor neurons is eliminated. They retract their presynaptic nerve terminal, which is quite an interesting phenomenon, and it deals also with electrical activity, synaptic activity. There's a competition between the two. It's very similar to what happens also in, for example, in the eye, where you have inputs from right eye and left eye, and you have to segregate those, and this is very similar also at the neuromuscular junction. You get this competition, and at the end you really end up with one single motor neuron innervating, one muscle fiber being innervated only by one single motor neuron. And also postsynaptically, you form these postsynaptic folds, which are important for this high efficacy of how to generate action potentials. Okay, once this is formed, you have a high concentration of acetylcholine receptors, and you may remember from neurobiology, acetylcholine receptors are basically consist of 5 subunits, 2 alpha subunits, 1 beta subunit, 1 delta subunit, and 1 gamma or epsilon subunit. And they form then a channel, an ion channel, and when acetylcholine binds to the alpha subunit, it opens, it gives a conformational change. There was just a Nature paper about 4 weeks ago where they showed the structure of the acetylcholine receptors and how the binding of acetylcholine then changes the conformation of these receptors and opens the ion channel so that the ions can flow. Ions that flow through the acetylcholine receptor are sodium and potassium, because there is this gradient. Sodium will rush into the cell, potassium will also rush out, but it's a much less of an electrical chemical gradient, so that potassium rushes out much less, but sodium rushes in, and this depolarizes the membrane. You reach threshold, and then the voltage gated sodium channels will kick in and completely depolarize the muscle fiber and generate an action potential. What you also see here, and this is important in the context also for this RNA fish experiment, that we have two different types of acetylcholine receptors. In the fetal or embryonic stage, the acetylcholine receptors are composed of two alpha subunits, one beta subunit, one gamma subunit, and one delta. And in the adult, the gamma subunit is not synthesized anymore and is replaced by an epsilon subunit, which then replaces the gamma subunit. The genes that encode the alpha are these CRNA1. This is how it's abbreviated, also so that you know in your RNA fish experiment you will do RNA fish for this alpha subunit, the gamma subunit, and the epsilon subunit, these genes. Alpha bungarotoxin, which you have also seen in the whole mounts, this binds to the alpha subunit and it's an antagonist to acetylcholine. It blocks the binding site for acetylcholine, and therefore if you are poisoned with alpha bungarotoxin, your muscles will not be able to contract and you will eventually die of respiratory failure. Okay. Now, these different acetylcholine receptors, the fetal and the adult form, they also have different electrical conductivity. The fetal form, these are patch clamp, single channel recording patch clamps. Here you see the opening of a channel and then the closing of the channel, and you basically see that they have different behavior. The adult channel has a high, this is basically this here, has a high conductance. It opens fast, lots of ions can rush through, but it has a short open time. It closes quite rapidly again, while the fetal form, which is this one here, has less of conductance, and it then closes later, only slower. This is a difference. It's still not really entirely understood what this difference here makes in terms of functionally. There were attempts to knock out, if you knock out, for example, the epsilon subunit, what then happens with a mouse, that they will be depleted of the acetylcholine receptor, and then in the adult stage, and eventually will be denervated, and then they start to re-express the fetal acetylcholine receptor, because that's what I'm going to show you now, what happens when you denervate the muscle. And this is also something you have in your mouse group, denervated muscles. When you basically lose the activity here, there are no action potentials generated anymore. The muscle becomes denervated, and then you look at the expression of these acetylcholine receptors, and these are Western blots of these acetylcholine receptors. You see here that in the innervated case, if you just mush up the entire muscle, you don't see much of these receptors, but they are only localized here at the synapse. No little alpha, little beta, no gamma, little delta, little epsilon. Now when you denervate, you see that you get a huge increase in alpha, beta, gamma, you get gamma, You also get lots of delta. You get a bit more of epsilon in this context. And now this is really driven by the lack of electrical activity, or this possibility to contract, because when you stimulate now with electrodes the muscle, then you start to see here that in the denervated and stimulated, that you still again lose the expression of the alpha, beta, gamma goes away, delta and epsilon is also down, which means that it's basically the electrical activity, the contraction of the muscle, that makes this extrasynaptic expression of the acetylcholine receptors. The same what happens also during development. Receptors are expressed there, and when you innervate, they are not expressed anymore in the non-synaptic myonuclei. They're only expressed in the subsynaptic or fundamental myonuclei when the nerve sits there. What you basically take home here is that the denervation causes an increase in the gamma subunit and all the other subunits in non-synaptic regions, and that it's the electrical activity of the muscle that decreases this gamma subunit expression in non-synaptic, in the entire muscle. So that only the adult form remains to be expressed at the synapse. And this seems to be mediated by calcium influx through these dehydropyridine sensitive calcium channels, the ones that are also important for the excitation contraction coupling, based on this stimulation here, and of course other experiments where you can block here these DHP sensitive calcium channels. Okay, these, as we also already mentioned, and you will also see this, some of these genes that are important to be at the neuromuscular junction are only expressed in these fundamental myonuclei, and are also then locally inserted into the membrane at the synapse. This includes the acetylcholine esterase. This is the enzyme that cleaves acetylcholine. You see here, this is like an old-fashioned RNA fish. It's basically done with probes that are radioactively labeled, and here you see a single fiber. It's actually a longitudinal section through muscle, and here you see the accumulation of this esterase. At these synaptic sites, there's also an increase in the epsilon subunit of the acetylcholine receptor message, also strongly increased, as you can see here. These silver grains, which are indicative of the radioactivity, are accumulated here. The same is true for the voltage-gated sodium channel, also accumulates here at the nerve muscle synapse in these fundamental myonuclei. Okay, this, which means the formation of the neuromuscular junction has two phases. In the first phase, it aggregates the acetylcholine receptors. Then when electrical activity kicks in, then there is downregulation of expression of these synaptic proteins like acetylcholine receptors in non-synaptic myonuclei, and it remains to be they remain expressed in these fundamental subsynaptic myonuclei, so that they can be locally inserted into the membrane. Okay, now the question is, what are the mechanisms that are important, and what are the molecules that are important during neuromuscular junction development? And I want to give you a bit of a historical background to this, how these molecules were discovered and how they were purified and characterized. One of the key experiments that was helping to identify one of the factors is schematically shown here. What you basically see here is a schematic view of a single fiber. In gray, this would be the muscle fiber. In brownish, in gray, you see the basal lamina, the basement membrane, the extracellular matrix that surrounds the muscle. I mean, laminin alpha 2 would be there in those things here. And here in green, you see the extracellular matrix that fills the synaptic cleft, which is called synaptic basal lamina, because it consists basically of components that are released from the nerve, from the muscle, I mean postsynaptic fundamental, and also from these capping Schwann cells. And then here in red you see the acetylcholine receptors that are aggregated at the synapse here, and this would be the presynaptic nerve terminal. Now what these people have done, that was done in the late 70s, they worked on frog neuromuscular junctions. They also have the same system as we do, and they injured at the pectoris muscle of the frog by freezing this, putting a metal piece that was frozen in liquid nitrogen and placed it on this muscle. And when you of course do this, you basically kill all the cells, the muscle fibers, also these Schwann cells, and you also get rid of the nerve here, that you damage the nerve, that presynaptic nerve terminal that sits here on the synapse. But you then end up, because after this damage, all the damaged cells, the dead cells, will be removed by macrophages that eat up these cell debris. You end up in a situation where the only thing that is left is the basal lamina, this basement membrane surrounding the muscle, but also this synaptic basal lamina is left, is not really chewed up by these macrophages. Now if you allow, and of course in frogs and also in mice, the nerve can regenerate, and if you allow now the regeneration of the nerve, but you prevent the formation of new fibers by killing the dividing satellite cells that are still here in this basal lamina sheet, but if you get rid of them by, for example, X-ray or by irradiation, then you will end up in a situation where the nerve grows back onto the original site, but there is no muscle fiber anymore. But what they observed is that the nerve that grows back looks actually very similar to the nerve that how it looked before of all this injury. Similarly, when they prevented the nerve from growing back by just cutting out pieces of the nerve, then the nerve cannot grow back, but they now allowed the regeneration of the fibers. They saw that these fibers, they also formed postsynapse-like structures, high concentration of acetylcholine receptors at the site where the nerve was sitting before. And the only interpretation of this experiment is that the structure that contains all of the information for both the pre as well as the postsynaptic part of the synapse must be contained in this synaptic basal lamina. And this was basically the information, or the key information, that then also allowed to eventually purify factors that would drive, on the one hand, post as well as presynaptic differentiation. But that's basically the conclusion what I already said here. And this is the experiment itself. Now this is a whole mount picture of a frog neuromuscular junction, you see, and here this is the acetylcholine receptors that are stained. Here would be the muscle, you see the striation a bit, and here is the acetylcholine receptors are labeled in black. And this is the situation what you have after this situation here, when the muscle is regenerated without the presence of the nerve. You still have you have, although it's maybe less nice than here, but you again have this aggregation of acetylcholine receptors here. You can imagine these experiments were done now 40 years back, and the techniques were still not were at that time, of course, not that nice as they are today, but maybe one can really see this nicely. This is now also a cross-section electron microscopic picture of such a regeneration, this as well, and I think this is the best picture here. These are acetylcholine receptors that have accumulated, and here you actually see still the hole in the basal lamina where the presynaptic nerve would be sitting, and here you see also these junctional folds that are filled here with this. This is also radioactivity staining for acetylcholine receptors. Even when you allow now the nerve to grow back without the muscle present, I mean this is this situation here, and this you look at also in the electron micrograph. This would be an electron micrograph of a frog neuromuscular junction. This here is the presynaptic nerve terminal. This is the capping Schwann cell. These are these vesicles that then fuse. This here is the postsynaptic muscle fiber with the fold. And when you see now in this situation, you have now a presynaptic nerve terminal sitting on an empty basal lamina. I mean, here you would be the muscle, but there is no muscle because this was prevented from regeneration. And you can really see nicely that this presynaptic nerve terminal looks like the nerve terminal, or quite similar to the nerve terminal, how it looked before the system degenerated. This really, as I said, is proof that what you have here is a signature in the synaptic basal lamina that allows the formation of pre as well as postsynaptic structures. And then, of course, you can formulate the hypothesis that the same factor or factors are responsible for the formation of the neuromuscular junction during development, although this, of course, is a regeneration experiment. But it's a bit similar to the denervation, reinnervation experiment. When you have in denervation, you have upregulation of embryonic forms of the acetylcholine receptor. It's a bit similar to the situation here. That's a hypothesis. It's not shown that this is really true. This experimental system that was then used to identify factors was cultured myotubes. These are cultured myotubes. You see here they are fused. They are green by the expression of one of the desmin protein, which is muscle myotube protein. And they also, when you culture myotubes, they express acetylcholine receptors. And they then tested, what they basically tested is the extracts from synaptic basal lamina to induce acetylcholine receptor aggregation. When you basically look at, you stain for acetylcholine receptors in muscle fibers or in cultured myotubes, they are more or less sometimes clustered, but not that heavily. And when you add extracts that then induce clustering, this is a sign of, like when when you co-culture the myotubes with motor neurons, you also end up in these clusters. And as a source they used, of course, not muscle, because in muscle you would be hard put to isolate enough of the synaptic basal lamina, at least at that time. They used an electric ray, torpedo, Californica. They generate a lot of current, and what basically their current generator is this electric organ. This electric organ is innervated by motor neurons of the electric lobe. But it's very similar to the situation in a muscle, but the electric organ loses all of the cellular contents. It's not something that can contract. It basically consists just of synaptic portions of these that are innervated by this electric lobe. This electric current that is generated is based on the fact that when these electric lobe neurons fire action potentials, then you generate a lot of postsynaptic current, and this generates a lot of high voltage, and this then allows these rays here to defend themselves when they are attacked by a predator. And this is also, by the way, the source of where people, as I said, have used to isolate acetylcholine receptors, the ones that were published about 4 weeks ago in Nature, the structure, they were also purified here out of these electric organs. This is really a very rich source of synaptic proteins. Okay, what they then did, they grinded up this electric organ basal membrane and then test it in this aggregation assay and then try to purify proteins that are active in this assay. This assay, of course, is not false proof, because there are actually quite a few things that also induce clustering of acetylcholine receptors. Latex beads is one example, probably not very physiological. Vitamin C, calcitonin gene-related peptide, and another protein which was called ARIA, which stands for acetylcholine receptor inducing activity. And I should mention, about maybe 20-30 years ago, these were all Nature papers. Each of this documentation was basically published in Nature, and it turns out that most of these things are, of course, not the ones that are holding the promise. The one thing that then still is holding is a protein called agrin, and you have also part of this is in one of your essays. I will emphasize then later that what you use there is not really the same agrin what you use here for this inducing of postsynaptic structures. It was purified based on its function, as I said, to induce acetylcholine receptor aggregation in cultured myotubes. It was then also shown that if you raise antibodies against this protein, that you can stain synaptic basal lamina. That's of course also an issue, because all the activities in the synaptic basal lamina. It also has been shown that you can deplete the extracts from this activity when you use these antibodies to pull out the protein, and also this antibodies against this factor can also inhibit the induction of acetylcholine receptor clusters in that are induced by the addition of motor neurons. These are important assays, of course, to show that this agrin is really also physiologically the factor that was localized in this torpedo synaptic basal membrane. The protein is a large protein, its cDNA is larger than 9 kilobases. It encodes an extracellular matrix molecule that contains domains that are very similar to other extracellular matrix molecules, and of course nowadays one needs to do knockouts of this protein in mice, and indeed when you knock out the protein, what you end up with a mouse that dies at birth because it cannot really form proper neuromuscular junctions. This is just a whole mount here, a schematic. What you get here, the nerve grows into the middle, but then does not stop growing, continues to grow, and acetylcholine receptors are not really clustered at the site where the nerves are growing. This is a whole mount picture of this at a rather early stage. You see here there are some clusters, but these are not the nerve branches out, and it's not really functional. Moreover, the basal lamina also seems to be disrupted. The structure has a problem here, as you can see in this electron micrograph. There was also another challenge, and I want to show that because I find this really a nice example of how splicing can affect biological function. I mean, as I said, this protein was purified based on its activity to induce acetylcholine receptor clusters, and here you see some of these clusters in a cultured chick myotube, and also based on the fact that antibodies stain the synaptic basal lamina. But there were also, when you use the same antibodies and you stain for kidney, lung, and other tissues, you actually also see that this protein is also expressed in non-synaptic regions. And when you grind up this lung and kidney and you isolate those proteins, or this protein that is immunoreactive here also with the synaptic protein, and you also test this protein in cultured muscle cells, there is no activity whatsoever to induce these acetylcholine receptor clusters. And this has raised the the discussion whether agrin is really the protein that induces here postsynaptic differentiation during development, or whether it's just some artifact and this here, and maybe there's an additional factor here that is important for this aggregating activity and that sticks to this agrin protein. It turns out that this is can all be explained by one particular splicing at one particular site, which is close to the carboxy terminal end of the protein, which is a site which is called B or C site. It turns out that there is really tiny alternative mRNA splicing occurring here at this site, which includes small amino acid inserts of 8, 11, or 19, which is 8 plus 11 amino acids that make this protein active in inducing clustering or not. I mean, here, this is what you can see here. These are just those response curves where you here measure acetylcholine receptor clustering per square micron, and you add different recombinantly expressed proteins that contain either this 8 amino acid insert here at the B site, or they lack this insert at the B site, which is here this one. You see, whenever you can add up to 100 nanomolar or 1 micromolar of this protein, it does not induce any clustering, while if it has this 8 amino acid insert here, it's sufficient. It really induces lots of clusters at quite a low picomolar concentration, which tells you, it's probably this splice site here that somehow is important, maybe to interact with its receptor to induce postsynaptic differentiation. But you also see here that you can make a very small version of the protein, which consists just of one of those domains here, the last laminin G-like repeat, and this is already also sufficient to induce clustering, as long as it contains here this 8 amino acid insert, although its efficacy is less than here. Okay. Which basically tells you that these splice versions really differ in their acetylcholine receptor aggregating activity. And as I said, this is when it lacks any amino acids here, there's no aggregation in those cases. If it has inserts, either 8, 11, or 19, 8 is really a very highly strongly active one, 11 is less active, and 19 is active as well, then you can induce acetylcholine receptor aggregation. Now, of course, you have to prove that this is really also essential for in vivo, and this is the experiment people have done to just remove these these inserts. The 8 and the 11 amino acid encoding insert are encoded in separate exons, which is here exon 32 and 33. And what they basically did, they created mice that lack these 2 exons by using LOXP sites and removing them in stem cells by using CRE, these exons, which then you're left with just one of the LOXP sites. These mice, they express agrin, but just they cannot include these C sites. And when you look at this, when they lack this C site, they are very reminiscent. That's actually the same phenotype as the full agrin knockout. You also have troubles to form these clusters, and they are not necessarily associated nicely with the presynaptic nerve. And also, if you were to wait longer, the nerve continues to grow and the mouse dies at birth because it cannot breathe. That's the first experiment that tells you that these sites are required for neuromuscular junction formation. The other experiment, this is an experiment we conducted in my lab then later, is basically use the knockout mouse and try to rescue just with this small version of agrin. And we basically used one that has a domain that is important for the binding to extracellular matrix, and just expressed here this small last G domain. And when you do that in a mouse that otherwise would die at birth, you can create synapses, as you can see here. And you also create mice that walk. This is just a brief movie of this. This is an agrin knockout which would die, and this is a 5-week-old mouse. I mean, it behaves very much the same as its wild type control. This experiment therefore shows nicely that expression of just this small version is also alone sufficient to prevent the neuromuscular junction failure, the formation of this failure. Just I want to briefly show you how we discovered then also the binding of this agrin to the extracellular matrix. It's an amino terminal domain in agrin called NTA domain. Basically, it's quite simple. We just measured binding of agrin to different purified components, and that time at the Biozentrum there was a professor who worked on these extracellular matrix proteins, and it was quite easy for us to do it because they had all these components purified. And you can see here that this agrin binds strongly to laminin, and even when you then make just this NTA domain alone, it's sufficient to bind to laminin, which clearly tells that NTA, this N-terminal domain of agrin, is sufficient to bind to the extracellular matrix. Therefore, what basically these experiments showed that as the hypothesis would have predicted, it's really agrin that is released from the presynaptic nerve terminal that then via some receptors induces postsynaptic structures, and this agrin then is immobilized in the synaptic basal lamina. And basically you need for this the very carboxy terminal end, the small domain, to induce, to bind to these receptors, induce the clusters, and you also need this splicing, while at the amino terminal end you need another domain by which agrin can bind to the synaptic basal membrane. And basically just these two domains are sufficient to fully restore the function of agrin to induce postsynaptic structures. Just as a reminder and an important remark, this agrin, what we use in these DYW mice, is actually the so-called inactive version of agrin, which lacks inserts here at the B site, because we realized that this agrin, without these inserts, binds very strongly to alpha-dystroglycan. And alpha-dystroglycan, this is basically shown here. These are alpha-dystroglycan binding of this 0 version. It binds very strongly to this, but it cannot induce aggregation, as I told you. Of course, we've used this portion of agrin that binds to alpha-dystroglycan with this NTA domain, so that we have these linkers between the laminin network and the alpha-dystroglycan. Of course, if you were to overexpress too much the neural version of agrin, you may end up having also troubles with the neuromuscular junction. If you can imagine, if you denervate the muscle and you express now agrin all over the place, you may confuse the nerve, and you may start to form postsynaptic structures where, because of the presence of agrin, whereas there's not necessarily the nerve. But just remember, this is not what you use here in the DYW mice, is not the one that has the proper inserts. Are there questions to any of this? Because then I suggest that we make maybe a 5-minute break so that you can breathe a bit and maybe get a coffee or something. Then we continue. Is this good? Please make thumbs up or thumbs down. Thumbs up, okay. At least few. Yeah, many. Okay, then we make 5-minute break, come back 20 minutes past 11. Okay, thank you. Okay. Should we continue? Are you there? Again, thumbs up if you're here. Okay, 1, 2, 3. Huh? Maybe some fell asleep. Yeah, anyway, let's start or continue. Okay. Now the question is, what are the molecules that respond to this agrin? And I want to give you a brief overview of work that was conducted maybe over the last 15 years or so, where people have defined the complex that is important for the response to agrin, which consists of LRP4, MuSK, DOC7, rhabsin, and the acetylcholine receptor itself. This is basically shown here, and over the years there was a protein identified which is called low density lipoprotein receptor related protein 4 or LRP4, and this was identified based on the fact that if you make a knockout of this protein, which was a protein of some function in skeleton, you get a phenotype which is very reminiscent to the phenotype of you observe in agrin. The same is true for this MuSK, which is a muscle-specific kinase. This is this protein here. It's expressed in skeletal muscle, and it has turned out to be the kinase that becomes activated upon the binding of agrin to LRP4, which then in turn activates MuSK. Again, this protein was discovered based on the fact that the knockout of MuSK creates a phenotype which is very similar to the agrin knockout. Then DOC7 is a protein also that is known to be, or a member of a family of proteins that are known to interact with receptor tyrosine kinases, and this protein, which is this small protein here, has been shown to interact with the activated form of MuSK and being important to keep MuSK active. It's like it's an abbreviation for downstream of tyrosine kinase 7 because it's the seventh member of this family. And it's important to keep MuSK active and also for downstream signaling to assemble the complex that is then important to maintain acetylcholine receptors there. Another protein which is not necessarily in this core receptor component complex is a protein called rhabsin. It has been discovered originally many years ago as being associated with the acetylcholine receptors. Rhabsin is this protein here. This is yet the other, this is the structural complex of the acetylcholine receptors that assembles with also the dystrophin glycoprotein complex, which at the neuromuscular junction is not the dystrophin glycoprotein complex, but the utrophin glycoprotein complex. Utrophin is homolog to dystrophin that is in adult situations only expressed at the neuromuscular junctions in the muscle, while dystrophin is expressed throughout the muscle. And dystrophin is the protein that is mutated in these Duchenne boys. To this complex also the voltage-gated sodium channel belongs, and many of these proteins you have encountered in the dystrophy lecture, which are these sarcoglycans, or also alpha and beta dystoglycan, which interact with utrophin. What is also seen here is the acetylcholinesterase, or its collagen-like form, which is the CoQ form, which is important to keep acetylcholinesterase in the synaptic basal lamina. But I will not talk about those proteins in great detail. I will talk about these proteins highlighted here mainly, and just briefly give you some insights as to how they were discovered and what their specificity is, and so on and so forth. Okay, rhabzin is a protein that, as I said, is associated with acetylcholine receptor clusters, with acetylcholine receptors, and when you add agrin to cultured muscle cells and you stain for the presence of the acetylcholine receptor, and you also see that esterases are co-aggregated, that there are also these different types of esterases are co-aggregated. But what is important, you also observe here aggregation of this at that time 43 kilodalton protein, which is called now rhabzin. This also associates with these acetylcholine receptors, which indicates that this must be part of this complex. And indeed, when you knock out rhabzin, these were really among the first knockouts done. That was in the beginning of the middle of the 90s. You see here that in the rhabzin knockout, these acetylcholine receptors. Now this is a whole mount on the muscle where you stain for acetylcholine receptors, and you see here that in these rhabzin knockouts the acetylcholine receptor clusters are completely gone. These mice also eventually die after birth because they cannot really breathe, which means that this association of rhabzin to the acetylcholine receptors is really essential also to hook it up to the complex that is important to keep the receptors in place postsynaptically. It turns out that this is based on this interaction of rhabzin with the acetylcholine receptor, which then in turn interacts here with eutrophin to keep it here in the place. Okay, the other protein, as I said, is one of these muscle-specific kinase, and again, as I mentioned already before, this was just discovered based on the fact that if you knock out MuSK, you get a phenotype in the muscle which is very, very similar to the phenotype in the agrin knockout. What you see here in these MuSK knockouts, you even have less of acetylcholine receptor clusters than in these agrin knockouts. This is really a classical example of a genetic evidence for a ligand receptor pair, and therefore at that time MuSK was considered to be the receptor for agrin. It turns out that when you express MuSK in cultured cells and you add agrin to cultured cells, and then I'm sure you know tyrosine kinases, when they become active, their tyrosine residues become phosphorylated. But when you measure phosphorylation of MuSK in response to agrin, there was never a phosphorylation of MuSK when you express MuSK, for example, in fibroblasts. Only when you express MuSK in myotubes, then it responded with phosphorylation to the addition of agrin, and which was argued as evidence that there must be a co-receptor to which agrin can bind and then mediate the activation of MuSK. But the knockouts are really very strongly reminiscent to an agrin knockout. Here you see the MuSK knockout. Again, you have presynaptic nerve terminals that are wandering over the muscle. They don't stop here, and there is no bungarotoxin. Also, this is RNA fish type of experiment. You see that the esterase is also not aggregated at the neuromuscular synapses. Now this co-receptor turns out to be LRP4. As I said, LRP4 is also a member of a quite big family of proteins, as indicated here. As I said, LRP4 is also expressed in the skeleton. There are mutations in LRP4 that cause skeletal defects in also human patients, depending on where they are. There are also mutations in LRP4 that cause problems at the neuromuscular junction, but when people knocked out LRP4, they really also saw the very same phenotype as they observed in the MuSK knockouts as well as in the agrin knockouts. And this, of course, triggered the idea that eventually LRP4 could be the receptor for neural agrin. Just when you compare that to MuSK, it's really quite similar, as I mentioned before. Then, of course, you need to show that LRP4 can bind to agrin, if it's were to be the co-receptor to which agrin binds. And this is basically shown here. These are simple fax experiments, and I mean the easiest way to look at this is maybe this here, where what you do here, you add agrin of different splice versions. Here this is adding LRP4, and then basically what you do, you add agrin, you measure the amount of agrin bound to LRP4, and here in this case LRP4 was added and agrin B0, this inactive version. You see you have only background binding. When you do agrin and LRP4, now agrin has this B8 insert here. You see there is binding, although it's not that strong. If you deplete of calcium, this is just a detail, don't need to remember this, but when you basically add LRP4 and agrin B8, you really get a very high affinity, strong binding of LRP4 to this B8 version of agrin, which would argue now this binding, in contrast to binding of agrin to dystoglycan, is really depending on on this insert at the site B8, which is quite reminiscent to, I mean, it's very good evidence that indeed this binding is the one that then activates MuSK. And you can also show when you now co-express LRP4 and MuSK in fibroblasts, and then you add agrin, then you can now also activate MuSK on this tyrosine residue, which is indicative of the activation of a receptor tyrosine kinase, as I mentioned before. Now, this has also then been crystallized, and what you basically see here is the crystal structure of the last LG domain of agrin with this B8, with the B or C8 loop in a co-crystal of the binding site in LRP4 to which this C8 loop binds. And you see here when you add these two components, the C8 loop here forms a quite nicely structured loop, and you can see that there are interactions with particular residues in this 8 amino acid insert here with this isoleucine and this arginine that interact with LRP4. These are also highly conserved in inserts. Here this is this isoleucine and here is the N. These are highly conserved residues in this 8 amino acid insert at this B site. Interestingly, when you just crystallize this last LG domain, here this is this chick agrin, and these are the crystal structures of chick agrin, which was done in collaboration here in the Biozentrum, and rat agrin, which is this work here. And this rat agrin is a co-complex with the LRP4 binding site. You see here that the chick agrin, which is this red structure here, that there is actually this C8 insert could never be visualized in the crystals because it was very flexible and was not really forming a proper structure, while the rest of the domain is very similar to the rat agrin, and only if it co-complexes together with this binding site in LRP4 you get this nice structure of the C8 or the B8 insert, which is really also again a strong indication that this is really the functional binding site to which neural agrin then binds and mediates its postsynaptic differentiation activity. Now there's this other DOC7 protein again, same idea. Here this is basically showing that DOC7 is localized to the synapse. I mean here these are cross sections. You have also seen in your immunostaining procedures, these are acetylcholine receptor clusters. This is a neuromuscular junction from the side in a cross section. That's how it looks. And you see here, when you stain for DOC7, it basically colocalizes with the acetylcholine receptor clusters. Here you see that when you overexpress DOC7 with MuSK, you get autophosphorylation of MuSK. This is often the case when you overexpress a tyrosine kinase with its adapter. Then you really get also autophosphorylation without the ligand of the receptor tyrosine kinase. And finally, as of course now you should obviously notice, when you knock it out, DOC7, you get a very similar phenotype as in the agrin knockout, in the LRP4 knockout, in the MuSK knockout. You get this presynaptic nerve that continue to grow, and no clustering of the acetylcholine receptors. Just to summarize this point again, all these knockouts, agrin knockout, this is now a later stage of the agrin knockout. You see here really also the strong nerve growth. These are LRP4 knockouts, DOC7 knockouts, and MuSK knockouts. This is unfortunately black and white. They really get the very similar phenotypes, and this tells us that these really form the core complex that is important for the formation of the neuromuscular junction. I should maybe also mention, if you denervate a synapse, agrin is the only one that is presynaptically released. All these other components are postsynaptic. When you denervate the muscle, MuSK expression is also upregulated in all non-synaptic myonuclei. The same is true for LRP4, and the idea there is that when you denervate the muscle makes itself ready for reinnervation. That's why these components that are important for the formation of the synapse then are increased in expression, so it becomes easier to reinnervate a muscle by the nerve that releases then agrin. If you could imagine, in an innervated case, you want to have no other nerve reinnervating the muscle, because you have only one motor neuron that innervates a single muscle fiber. That's why you have to make the non-synaptic region non-responsive to reinnervation as long as the nerve is properly functioning and generates this electrical activity. That's why you want to get rid of all of these proteins that could make then synapses to work. And only when you denervate, you want to make the muscle again responsive for all of this agrin that is released from presynaptic nerve terminal. Of course, when you think of diseases that affect any of those molecules can of course also cause problems in neuromuscular transmission. And indeed, when you see here these proteins we were discussing, like DOC7, MuSK, agrin, they are all also observed in some of the diseases that cause congenital myasthenic syndrome. These are genetic diseases where the patients have difficulties in neurotransmission. And you see here the frequency is actually quite low for agrin, MuSK, and DOC7. There are more cases of rhabsine and more cases of acetylcholine receptor mutations. Can someone think of reasons why this is more frequent for the acetylcholine receptor and for rhabsine than for those other candidates? If you think of the phenotype of the knockouts here.
If any of the other one is damaged when the phenotype is lethal?
Yes, exactly. I mean, this here is often lethal if you have a null mutation. The baby will not survive. Well, if you have a mutation in the acetylcholine receptor that is maybe making it less functional, you can still survive, and that's why these phenotypes are then also observed in the adult. This is exactly the reason. I mean, the agrin mutations that have been documented so far are mutations that affect regions in the agrin molecule that are not necessarily have been shown to be 100% involved in acetylcholine receptor aggregation. They may make the protein hypomorphic. There is less agrin. The same is also true for mask mutants. They never affect the essential tyrosine that is important for its function, of course. Okay. Now, just briefly, these molecules are also okay. Now, one issue was always why do we also have this presynaptic effect? One of the things that were speculated is, of course, when you don't have onset of electrical activity, you don't have aggregation of the postsynaptic structures properly, and the nerve continues to grow because it cannot really form, because these accumulations of the acetylcholine receptors are not sufficiently high. It turns out that one of the candidates that could act on this is in fact LRP4, and I just don't want to go into the great details, but I want to show you what the approach was people did. When you have a global knockout of LRP4, as I told you, this is this mutant here, you have a very strong pre as well as postsynaptic phenotype. Now these people have then started to knock out LRP4 conditionally only in muscle by using a Cree mouse that drives Cree only in skeletal muscle. And what you see here is that you still get some acetylcholine receptor clusters, and the sprouting response is less pronounced than in the complete null mouse. If you do the same, when you just knock out the LRP4 in motor neurons, these are HB9 is one of the factors that can be used to drive Cree selectively in motor neurons, you get actually almost no phenotype. But when you combine HSA and HB9 knockout mice, then you get a phenotype which is very reminiscent to a global knockout. And this argues that LRP4 released from the motor neuron might also be a factor that helps the motor neurons to stop and to form a presynaptic nerve terminal. Another way to show this is by culturing motor neurons with myotubes. When you do this, motor neurons that encounter acetylcholine receptor clusters, they form also presynaptic nerve terminals. Now you can replace a myotube by a fibroblast that overexpresses LRP4, and when you do this, this is basically shown here. These are 3T3 cells that overexpress LRP4. You see that when you overexpress LRP4 in these 3T3 or 293 cells, you get also some presynaptic nerve terminals that are, you have aggregation of synaptic vesicles, and this would tell you that this is also LRP4 somehow feeds back onto the presynaptic nerve to induce presynaptic differentiation. And this is basically the model how people would argue this happens. Whether this is true is not entirely clear. Whether there are other factors could also well be, but at least this is a candidate, and I just wanted to show you this evidence quickly. Finally, as I said, these molecules are also important for the maintenance of the neuromuscular junction in the adult, and the experiment to be done is that if you knock out agrin only in the adult situation, when you do this is by inducing also a conditional knockout in motor neurons. And if you do this, you end up after 3 months, you see here that the synapse in the adult, after 3 months of knocking it out, you lose presynaptic nerve terminals at postsynaptic.
Excuse us, the microphone stopped working. We can't hear you anymore.
Now, do you hear me? Yes. Okay. What was this? Okay. I'm sorry. Now, did you hear this? This you still? I mean, this is basically the same experiment. You knock out mask in the adult, and you lose also innervation. The same is true for LRP4. This is done with an inducible. Uh-oh. Now everything crashed. You cannot minimize. Okay. So, sorry, will eventually come. Don't want to have screen on. Sure. Okay. Come on. Okay. Sorry. Okay, you see this again? Yeah. Okay, this is basically the experiment with DOC7 where they used siRNA to knock down DOC7, and what you basically see here that the presynaptic and the acetylcholine receptor positive area get smaller with time. They didn't wait until you get a complete innervation, and siRNA is maybe also not removing the protein completely. But this is basically also what you observe here, that when you knock down DOC7, you also start to denervate the muscle, and you lose postsynaptic differentiation. Okay, this is basically the take-home message then. For this part is that this splicing, what we discussed, the acrin receptors, there is mask that is necessary for the signaling, LRP4 that binds to the active version of acrin, DOC7 that is required to activate events downstream, and for hooking up to phosphorylated mask, rhabsine that serves an adapter to bridge the acetylcholine receptor to the cytoskeleton. And I also showed you some evidence that LRP4 might also be a factor that signals retrogradely to the presynaptic nerve to induce presynaptic nerve specialization. And finally, I also showed you that all these proteins are also required for the maintenance of the neuromuscular junction. Now at the very end, bear with me, it's not going to be maybe only really just 10 minutes. I want to briefly also show you this aging situation, and I want to emphasize one of the proteins that seem to be important. I discussed this already with you, that there are many different possibilities how aging can be affected in skeletal muscle, and that these are these hallmark criteria of aging. And I just want to discuss whether mTOR complex 1 is involved in sarcopenia, because it's something that we think is really the case. But I want to sort of elaborate to you what the different considerations might be that one needs to consider when one discusses mTOR complex 1. Also, you have these discussions on muscle sizing. That's why I would like to start with this. I mean, I've shown you this, that protein synthesis is important for muscle hypertrophy, and unloading of the muscle causes muscle atrophy, and protein degradation is important there. And it's very obvious, or it has been shown by several groups, that it's really the IGF-1 AKT mTOR FOXO axis that is important in this context. And it has been shown when you block mTOR complex 1, and you probably know that rapamycin or rapalogs are inhibitors of mTOR complex 1. When you do this, when you do such an overload experiment in the presence of rapamycin, there is no increase in muscle size. This is shown in this old paper here by the group of David Glass at that time. What they basically showed here is that you can compensatory hypertrophy is a way when you unload your muscle, and then you get an hypertrophic response when you load the muscle again, and this is called compensatory hypertrophy. When you do this in the control or with here, you get an increase in the fiber size. But when you do that in the presence of rapamycin, you do not get any or not much increase. It's not significant compared to not in hypertrophic responses here in the presence of rapamycin, which means it's really mTOR complex 1 activity is essential for inducing muscle hypertrophy. And as I said, and we also look at raptor knockout animals in some of your experiments, and I've told you in the beginning that raptor knockout animals, they also show some issue with muscle atrophy. In other words, this protein or this complex is important to make muscle bigger. The trouble with this is that there are also other observations that concern aging. I told you one of those is that mTOR complex 1 is active in aged muscle. This you can see here. This S6 phosphorylation is increased. 4-EBP phosphorylation is also increased, which are two of the downstream targets. You see that here and here. And the same is also true in humans, and this argues that why would you increase mTOR1 activity at the high age in a situation where you actually lose muscle mass instead of increasing muscle mass, as you would argue in a hypertrophic response where you activate mTOR complex 1. The other observation that is striking is when you treat mice with rapamycin for quite some time and you measure survival of these animals, and this was a Nature paper published also 10 years ago now, and these rapamycin treatment mice are the ones with the red dots here. You see that rapamycin treatment mice, and these are different sites, University of Texas, this is University of Michigan, and this is Jackson Laboratories. They really treated many, many mice, and you see here that in males, and even more so in females, the treatment with rapamycin prolongs lifespan. I mean, the median survival of these animals is clearly longer in the presence of rapamycin than without rapamycin. The same is true when you knock out one of the downstream targets of mTOR complex 1, S6 kinase 1, you also get a prolongation of lifespan in those animals, which would argue that dampening, lowering mTOR 1 activity would increase lifespan. But now in the context of muscle, of course you ask yourself, but rapamycin also inhibits hypertrophy. What would this then be doing to the muscle? Maybe the muscle becomes smaller in the presence of rapamycin in these animals, and despite these small muscles, animals would survive longer because they may be less prone to get cancer or whatever, something that shortens your lifespan. And therefore the question is, what is the effect then of low-dose rapamycin treatment on the muscle and on the neuromuscular junction? That was a question we were addressing, and in particular Dan was actually addressing this question. And we did this, which is quite a dragging experiment, you can imagine. We treated basically mice with low dose of rapamycin in the food for quite some time. We started at 15 months with mice and then examined them during those another 15 months until the age of 30 months. And some of the cohorts we treated at 20 months of age and also looked at them at 30 months of age. And now when you just look at the body mass, and these are the two curves, you see that rapamycin lowers overall body mass, and you see that in the control here that the aged mice also start to lose body mass at a certain age. But what you also see is that irrespective of when you start treating the mice, both of these rapamycin treated mice, they end up with a very similar body mass. That's why we combined the two cohorts and then measured the outcome at the end at 30 months of both groups combined, which gave us quite a high number at 20 mice here and over 20 mice in the rapamycin treated cohort. When you just measure certain aspects of function, muscle function, and these are all the rapamycin treated mice here in reddish, you see, for example, grip strength, which is a measure of muscle force, is improved by rapamycin. Running is improved by rapamycin. If you put them on a hang test, it's improved compared to the 30 months old mice. These are the 10 months old mice, which are still stronger, but they lose much less of the grip strength. They also, when you measure in isolation the force generated in soleus muscle, you see that rapamycin also basically prevents the loss of strength at 30 months. But importantly also, when you look at skeletal muscle mass over body mass, and these are different muscles we have looked at, it's quite interesting to see that some of the muscles, like triceps muscle, which is a forelimb muscle, you also look at in the DYW mice, this muscle responds very well to the rapamycin treatment, which is this cohort here, while in high age it loses mass. This loss of muscle mass is completely rescued by the presence of rapamycin. In stark contrast to this, the gastrocnemius muscle, which is another hind limb muscle, loses also quite some mass at high age, but this mass loss is not at all prevented by rapamycin. In other words, what you see here is that most muscles respond to rapamycin, but some muscles don't respond in terms of the muscle mass preservation to rapamycin, which again tells you a story about differences between different muscles. But what you also observe when you look at the neuromuscular junctions, that this rapamycin, when we look at 30 months old mice, that the rapamycin treated mice, their neuromuscular junctions look actually quite similar to the neuromuscular junctions at 10 months old controls, unlike the 30 months old mice where we start to see denervation effects and thinning of the axon. This is just a quantification of some of the effects that you see here: preservation of axon diameter by rapamycin. You see less of the nerve sprouting, and you also see that fragmentation is much less in the rapamycin treated animals, which would argue that indeed one of the hallmarks is fulfilled also with mTORC1, that when you attenuate the pathway, that you can slow many aspects of aging. Okay, this is then the conclusion, that dampening of the signaling here improves not only the lifespan, but it also improves muscle mass and function. And I also told you or showed you that different muscles respond differently to this rapamycin treatment. Now the other question we addressed, and also this is one of the mouse models you have in your aging group, is a mouse where we exacerbated the pathway by activating constantly the pathway. These are these TSC mice, what we have discussed already, where you have this difference in proteasomal degradation and protein synthesis, what you also observe in mouse group 2. And we basically just wanted to see whether this could be a good model that for sarcopenia. And as you now probably know, indeed, it seems to be quite a good model, because what we observe here is quite some changes of the neuromuscular junction. And since these TSC knockout animals, they age faster, we observe these differences already at 12 months. As you can see here, these are the neuromuscular synapses. They look very much more fragmented, and also some sprouting effects we observe here in contrast to the control situation. And some of these aspects can quite interestingly be quite improved by a short-term, only 4-week treatment with rapamycin. Again, you can quantify some of these aspects and compare that to the situation at high age. Many of these features that are otherwise only observed at 30 months old control mice can already be observed at 9 months old TSC mice, like this axonal diameter difference. The nerve sprouting is a very strong response in the TSCs, and also this the extent of fragmentation is well observed. And many of these, although not all, can be reversed by, just as I said, 4-week treatment of rapamycin of these TSC effects, while some of these aspects cannot be restored, which is maybe a secondary consequence of the activation of mTOR complex 1. Finally, we also have measured function at these synapses, and what is interesting here in these TSC knockouts, that we see differences in the postsynaptic currents. These are these two numbers here. You see the knockouts have higher postsynaptic currents and a higher quantal content, which is quite interesting in the context of aging. That would argue that these synapses, because they have some troubles, they start to compensate presynaptically by releasing, for example, more neurotransmitter. But whatever the response is to this, we will look at this now in greater detail. But it's interesting to note that when you look in old mice, there are these parameters like postsynaptic current as well as quantal content, although it does not reach significance here, is also increased in these old animals, which would argue that indeed the TSC knockout animals are quite a good model for premature aging. Finally, we also get loss of acetylcholine receptor density, which is also a sign of aging, and what we observe here in these in the TSC animals, we observe quite a strong loss of acetylcholine receptor density. At least partially it can be reversed by rapamycin, and this is also observed in aged animals. Okay, this is basically leads me to the conclusion that exacerbation of the pathway also really accelerates aging in the muscle and causes precocious sarcopenia. And this is for us now a sign that we can model early aging, and that this mouse model might be really a good mouse model to test the function of some candidate genes we have now discovered, and also some pathways we would like to examine whether they have any effect on the aging process. And because, of course, you can imagine this is something that is really of interest to the society as well, to understand better why in age we lose muscle mass. Okay, with this I'm done, only 5 minutes late. Are there questions? If you're not too exhausted, I'm exhausted too. Okay, seems not to be the case. There's someone connecting. Maybe not. Okay. If this is not the case, I wish you good lunch, and then we'll continue at 1 for most of you. Thank you for your attention.